Hello. My name is Kambiz Bazari. I'm a practicing orthopedic surgeon in Northern California. My company develops tools, techniques, and implants that make surgical procedures safer for patients. I'd like to discuss an unsolved problem in total hip replacement surgery. This surgical procedure is a $7 billion industry. It's estimated that 13% of hip replacements fail within the first 10 years, and 75% of these failures occur due to aseptic loosening. Aseptic loosening occurs when a surgical implant fails to bond to bone. These failures cause pain, suffering, and loss of mobility for the patient. When a hip replacement fails, a revision surgery is performed to correct the problem. These revision surgeries cost up to $2 billion per year, and the results are rarely as good as a primary procedure. Up until now, the implant itself has been blamed for aseptic loosening. However, many believe that the surgeon's assembly technique is actually responsible for these problems. I'm developing alternatives to the primitive tools that surgeons are forced to rely on. With these new tools, the surgeon will have an alternate assembly technique that will prevent aseptic loosening and the need for revision surgery. Orthopedic surgeons use a technique of bonding implants to bone known as press-fit fixation. Central to this technique is a surgical mallet, which is used to repeatedly impact the implant into the bone cavity. The goal of this technique is to obtain optimal stability of the implant through interference fit. Interference fit is achieved when the implant is fully grasped within the bone cavity. This occurs when the implant is properly sized and is neither too tight nor too loose. However, finding the proper size of the implant is elusive because it can only be determined qualitatively through the surgeon's auditory and tactile senses. Only by listening for the changes in pitch and feeling for the changes in the bone's grasp of the implant can the surgeon know whether they've reached optimal implant stability. Most surgeons never develop the qualitative senses they need because 80% of surgeons who perform total hip replacement perform less than 10 per year. When a surgeon performs a hip replacement, they must first size the bone cavity by broaching or reaming the bone cavity. These bone cavities have certain physical properties and behaviors. When stretched, they behave like springs and produce a reactive force that grasps the implant. These physical properties can be modeled with a classical stress-strain curve of material science. Each cavity is unique because each patient has a different bone mineral density based on age and sex. And all bone cavities have their own distinct elastic limit or sweet spot of optimal stability. This is the point at which the cavity can no longer stretch to fit the implant. This limit is known to be between 0.5 millimeter to 1 millimeter. Past this point, and the cavity fractures. Conversely, if the bone is not stretched enough, then a tight fit is not obtained at all. In this sizing process, the first error is introduced. Error is also possible when force is applied to install the implants. At this stage, the surgeon uses a surgical mallet to repeatedly strike the implant into the bone cavity. And here, the surgeon has no idea how much force they're applying with each mallet strike. Mallet strikes vary wildly in their amount of force. And this compounds the original sizing error by causing occult fractures, damage to bone cells, and bone vascularity. Considering all this, it's clear that surgeons do not have the proper tools to press-fit implants reliably. I'm developing a set of assistive technologies to help surgeons press-fit implants properly. These technologies provide a quantitative method of sizing bone and a controlled method of applying source force to install implants. These technologies will prove to be safer, more efficient, and in time they will become the standard of care. These technologies are premised on replacing the surgical mallet with an alternative. Force applied by the mallet is problematic for two reasons. The mallet gives the surgeon no control over either the magnitude or the direction of force that they apply. When the magnitude of force is unregulated, the surgeon can either over-impact or under-impact the implant into the bone cavity, both of which can cause aseptic loosening. When the direction of force is unregulated, the surgeon can cause undesirable torques with each mallet strike. This can cause malpositioning of the implant, which can also necessitate hip revision surgery. To replace the surgical mallets, I'm developing vibratory insertion tools. These tools will utilize both sonic and ultrasonic vibrations. This technology for implant installation has a distinct advantage. It disarms frictional forces at the implant-bone interface. This allows easy control of alignment and requires much less force for implant installation. When less force is applied, bone cells and bone vascularity are not damaged, and when frictional forces are dramatically reduced, surgeons can continuously adjust the alignment of the implant. This type of adjustment is not possible with the surgical mallet. With each mallet strike, the implant is locked into position with no possibility of adjusting the implant between mallet strikes. The development of vibratory insertion technology is also critical to the development of next generation of implants. Many future implants will have delicate sensors to detect infection and metal debris, and the high magnitude impacts of the mallet may damage these sensors. Vibratory insertion tool will also incorporate IMU technology, and IMU display will provide the positional readings of the implant's alignment. This will give the surgeon the opportunity to make real-time adjustments. Even with all this functionality, this tool will be light, handheld, and will not add cognitive load for the surgeon. To accompany the vibratory insertion tool, there are two systems that can size bone cavities quantitatively. The first system uses a sensor suite within the bone preparation tool. This suite measures forces both within the bone and within the tool itself. These measurements are fed into a feedback algorithm that signals to the surgeon when they've reached the elastic limit of bone. The second system utilizes analog electronics to measure current and power consumption. These variables have a direct relationship with the frictional forces that exist at the implant bone interface. This relationship can be utilized to quantitatively identify the elastic limit of bone and therefore the sweet spot of optimal stability for each patient. The immediate market for the vibratory insertion tool is the global total hip replacement market. The eventual market will expand into knee and shoulder arthroplasty and then into trauma-related implants. What we're talking about here is standardizing the assembly technique in orthopedic surgery. We don't know of any other company that's thought of this endeavor. Once these technologies are developed, I anticipate licensing them to the major medical device companies. I anticipate these tools will easily pass 510(k) pre-market notification. The commercialization phase will also involve collaboration with these companies to manufacture, market, and distribute the tools. The basic tool itself would likely cost no more than $2,000 to produce. It will have a durable lifetime of 2 to 3 years or an expanded lifetime with maintenance and service contract. In the United States, there may be up to 10,000 hospitals and surgery centers that may each make a capital purchase. The tool will be open source, allowing for use by all medical device companies. This tool presents opportunity for additional revenue because it requires a disposable adapter for use with various implants. Disposable adapters can be produced at a low cost and retailed at a high premium. The orthopedic disposable market is a $59 billion industry. I'm working with academic researchers and contract engineers to develop these ideas. I'm seeking $4 million in funding to finish development of these tools. With this funding, these tools could be ready for clinical testing within 18 to 24 months. Thank you very much.